you will use a dendritic cell as a target, and this dendritic cell will present the antigen that you want in a way that any T cell that is specific for that antigen that you are giving the antigen presenting cell will proliferate, and then you can count these cells. So comparing to the other methods of detection, it will be an indirect method. It's not direct as the monomer, but it will not be restricted to a single HLA or a single antigen. You can give the dendritic cell a whole pathogen, for example. And that was exactly what Federica Saluso has done in 2009. She developed a method that is called the T cell library, which is a method to comprehensively evaluate the antigen-specific T cell repertoire. How does she do it? So she basically sees the T cells, and these T cells can be pre-sorted. For example, you just sort the naive T cells or the memory T cells, and you expand those cells in a way that later you can use autologous monocytes with different antigens to see how many T cells are proliferating in each well. And she uses a kind of complex mathematics with Poisson distribution just to calculate the frequency of cells in the ends that were initially at the well that were specific. So by doing that, you can see here, for example, when you use libraries of naive central memory and effector memory cells, and you use the tetanus toxoid against these libraries, you will see that these patients that had already contact with this antigen has T cells that are specific for this antigen on both the naive, but also on the central memory and effector memory compartment. While if you use another antigen that this donor shouldn't have contact with, you will only find T cells in the naive compartment because they did not have the contact with the antigen, and you don't have any memory cells. So when I saw that, that actually was a work that she presented on one of the society of the Brazilian Society of Immunology conference, right when I was finishing my master. I was very interested to try to see if we could do the same thing with cancer cells and with tumor, because so far it's very, very difficult, as I mentioned, to calculate the total tumor-specific T cells, especially if you don't know what is the antigen to look for. So what I have done during my PhD was actually I went to her lab, and we actually had to modify a little bit the protocol to be able to look for frequency of tumor-specific T cells. And that was exactly what we have done. So we swapped the monocytes to monocyte-derived dendritic cells because they are more potent. And we have done a couple of other modifications to be able to look into the frequency of tumor-specific T cells. So when we did the same thing in healthy volunteers, it's quite interesting that, of course, we do find tumor-reactive T cells in the naive compartment, but we can also find tumor-reactive T cells in the memory compartment in some healthy donors. So that is a known thing that is called tumor immunosurveillance. So, of course, we don't know if these cells are cross-reactive to an antigen that is expressed on the tumor cell, but they are in the memory compartment, so they can potentially surveil the body and find cancer cells before they become tumorigenic. And that's the main thing, because we know now for a long time that people with HIV or even patients that are under immunosuppression because of a transplant, they do develop more tumors later in life, and that is probably due to not having those tumor-reactive cells in there working on their favor. But we also wanted to look into cancer patients, which was the most interesting part for us. So we know already that the tumor can have very different immunogenicity depending on where it grows in the body. So here, for example, is just a number of somatic mutations per megabase, which gives an indication of how much of those neoantigens that each tumor will have. And you can see how varied it is depending on where the tumor is growing. But not only that, but you also have difference in expression of the tumor-associated antigens. So you have tumors that express many types. For example, the melanoma express a lot of cancer testis antigens and also other classes of tumor-associated antigens. And it's also one of the ones that have the most mutation burden, which means it will have the most number of neoantigens. And what happens now is that we know that the more immunogenic tumors they have higher response to immunotherapy. And there are many antigens that are already known for these classes of tumors. While for the less immunogenic ones, we have patients that have poor response to immunotherapy, and usually the antigens, if there is any, they are still unknown. So the more immunogenic ones, usually the ones that are most studied are the melanoma and the lung, and these are the ones that have the highest response, for example, for the PD-1 and the anti-PD-L1 treatment. But on the other side, you also have, for example, the breast and the glioblastoma, which are not very immunogenic. And what we wanted to know is, can we identify tumor-specific T cells in patients with less immunogenic tumors? Because so far, the literature was considering those as quite cold tumors, that they wouldn't have tumor-specific T cells, and that was why they did not respond to immunotherapy. So we wanted to be sure that this was actually true. So we used that protocol that I mentioned in both healthy patients and patients with breast cancer and glioblastoma, and calculated the breast tumor-reactive T cells for both the CD4 and the CD8 compartment. And as you can see, in the healthy donors, as I mentioned, we do have those cells in both the memory and the naive compartment, but we do have also these cells on the breast cancer patients and the glioblastoma. But the glioblastoma is way lower than the breast cancer. So that kind of patient, we would think that if it has any antigen, and a potential antigen, it might benefit for a vaccination, because they don't really have a lot of tumor-reactive T cells to start with. So if we could potentially vaccinate these patients and increase this number, that would be beneficial for these patients. But the problem is, like, can they really have tumor antigens? And do they have this potential? So I was very lucky that when I was in the PhD in Barbuto's lab, he was developing this hybrid cell clinical trial in glioblastoma patients. So this is basically a vaccine trial where you use dendritic cells from healthy donors and tumor cells from the cancer patients, and you fuse those cells in order to have the HLA typing of the tumor cells, but also the full potential of a dendritic cell from a healthy donor. And we had a very, very interesting case report from a patient that already had 2 surgeries. Actually, this was the 3rd surgery that the patient was having to remove a brain tumor. So this is a very aggressive tumor. It's a glioblastoma. And these patients, they usually don't survive for very long, especially if it's already like the 3rd time that their tumor is being removed. But what the surgeon realized when we started doing the vaccination, so the vaccinations were being done every month, once a month, was quite improved in the image here, but also on the patient. So the patient was not, so these tumors are, as you can think, they can affect a lot of the functions of, walking, talking, all those things. And this patient had already like a low function when they started treatment, but they recovered for a while until the tumor came back. And unfortunately, the patient died after a while. But this was a quite striking recovery. And we were asking, can we understand what was going on with the immune response and what was the vaccine doing during this whole period of treatment? So we have done the same analysis on this patient during the different time points. So this is what happened before the treatment. So we were not able, so this assay that I mentioned has a limit of detection. So we were not able to detect under above the limit detection of the assay any CD4 and CD8 before the treatment. But we could still do the analysis on the phenotype of the CD4 cells, because we do add IO2 after we do this analysis. We add IO2, which is a cytokine that increases the proliferation of the cells, and we keep the cells 2 more days until we do the analysis of the phenotype of the cells. So we can still do the analysis of the phenotype. And when we analyze the phenotype of these cells, these tumor-reactive CD4 T cells, you could see that there is a lot of cells that are IL-10 producing, so those are Treg cells, and not as many Th1 cells. So this is just removing the Th0, which would be the cells that didn't express any cytokine. You could see the percentage of each of the different subtypes of the CD4. So what happened when the patient started treatment? Quite strikingly, the CD4s went up first. On the very first dose, it was already up. The CD8s took a little while, but it also went up after the CD4s, which is expected because we know that the CD4s they helped the CD8 response. But quite striking, not only the response was up, and we could now detect, but the phenotype of the cells completely changed. And now they were mostly Th1 by those three. So, and the Tregs were completely gone. What was quite interesting was like these patients remained well for a while, but then the tumor recurred, as I mentioned, and we could see a drastic change right before recurrence for this tumor, for this patient. So there was a drop in the CD4s, and the CD4s became producing of IL-17, which is quite interesting and show how this method of analyzing the tumor-specific response can give you hints on what is going on with the patient, and it could help develop better treatments once we know what we want and what the treatment is doing. So moving on to my postdoc now, I was very interested on the breast cancer, and I wanted to go back to them because it's a cancer that is very, very prevalent. And basically 1 in every 8 women will have breast cancer during their lifetime, which is quite high. And we have, for example, more than 200 new cases and 52 deaths per day in Brazil for breast cancer. So it is a very big concern and still not solved, because we still have a lot of patients dying from this kind of tumor. But this breast cancer is very heterogeneous, and it is divided into different subtypes. So we mainly have the division is due to the expression of 3 different molecules. So it's the receptor of estrogen and progesterone, ER and PR, and HER2. So if the tumor expresses any of the hormone receptors, so we call them hormone receptor positive, which is 70% of the cases, they would express estrogen receptor and progesterone receptor. While we do have the HER2 enriched, which overexpress HER2, and then you have the triple negative that doesn't have expression of any of these markers. And they are quite different by itself. First, the prognosis. So you have best prognosis on the hormone receptor positive, but also on the way that you treat. So of course, for the hormone receptor positive, you can use things that block the estrogen receptor or the estrogen itself to treat the disease. For the HER2 enriched, again, you can also have targeted treatments with antibodies anti-HER2. But for the triple negative, there is no target treatment, but you do have the immunotherapy as a way of treating. And that's because the response to immunotherapy increases as you go with the worst prognosis subtypes. And this is actually has been FDA approved, although the response rate is lower than other tumors. And this is actually goes hand in hand with the presence of tumor antigens. So we know that the triple negative has more antigens than the hormone receptor, although even the triple negative doesn't have a lot of antigens. And what most people think before was, okay, so luminal A and luminal B patients, they don't respond to immunotherapy, and they would think is it due to the lack of antigens? And that was what the field would think before, because they couldn't identify any antigen specific for these cells. So we wanted to ask, is it really due to the lack of antigen? And can we identify better immunotherapeutic approaches to use? Because as you can see, although this is the one with the best prognosis, it accounts for 3/4 of all the patients. And once they have become metastatic, it is difficult to treat. So that was the work that I will show you now, which was published last month in Cell Reports magazine. So what did we want to do when we first started the project? So first, we wanted to identify the CD8 tumor-reactive T cells in the blood using that method that I mentioned. We wanted to evaluate the TCR repertoire, the diversity, the overlap of these repertoire, because we do have capacity in here to do TCR sequencing and all these analysis that was not possible during the PhD. And we also wanted to look for specificity. So what are these cells specific for, and confirm if they are tumor-specific. Of course, we wanted to correlate what we were seeing on the circulation with the response on the tumor, and also look into the clinical characteristics of the patients and how that correlated with the presence of the tumor-specific T-cell response. So first of all, we started with a very similar method. So from the blood, we collect the monocytes that we differentiate into dendritic cells, and we collect the T cells that we stain with the CFSC. And we co-culture those cells. So we load the monocytes with autologous tumor lysates from the patient. So that will contain all the proteins that are present on that tumor. So this means that we can look at the full tumor-specific response, even though we don't know what antigen is expressed on that tumor. And then we do a co-culture and a flow cytometry where we see the dilution of the CFSC, which means that the cells proliferated, and the CD25 expression. And what we saw on this cohort of hormone receptor positive was that 70% of the patients, they did have this response to the tumor. So we wanted to see, are these cells really specific and able to kill tumor cells? So what we did next was to do a single cell sorting and generate and expand tumor T cell clones. Because each T cell will have a different TCR, we wanted to test if we have different cells that are able to kill the tumor cell. So this is just one example of two different clones from the very first patient. Clone 1 was reactive to MDA-MB231, and the second clone reactive to HCC1937. So it's very difficult to grow the autologous tumor cell. So we end up using cancer cell lines to prove the specificity of the cells. So we use cancer cell lines that have matched HLA typing with the patient, and you can see how these, clone 1 react to a different cell line than this one, and those have different HLA shared with the patient. Just to confirm, for example, that the MDA-MB231 was HLA-A2 restricted, which was the allele that was shared within the patient and the cell line. We just did a blocking assay, so we use IgG control or blocking the full MHC class 1 or only the HLA class 2, and you can see HLA-A2, and you can see how it's blocked when you have either the antibody for the MHC class 1 or the HLA-A2, confirming the specificity of the cell and that it was restricted by HLA-A2. So of course, we also wanted to look into the killing. So this is killing assay using microscopy with the InfuSight machine. So you can see the increase in the green intensity means that the cells are dying. But we have also done a flow-based assay that show the same thing. So the cells can kill and they can recognize tumor cells. So next, we wanted to look into repertoire of the tumor-specific repertoire. So the sequencing of the TCRs of these tumor-reactive T cells. So we managed to find around 10 to 500 different TCRs per patient, depending on the patient. And to look into the diversity, what we use, so just a little bit of how the TCR is generated, just as to remember, because that's important for the analysis. So we have the VDJ fragments in the genome that recombine to create the single TCR. So what we have in the end is a TCR that has used 1V, 1D, and 1J for the beta, and 1V and 1J for the alpha. The alpha doesn't have the D. And these sequences, they are different. So you can, for example, look into the diversity by just looking at the pairing. So what V gene and what J gene each of the TCRs used. So that is exactly what we did in here. So you can see the different clones. Sometimes you do have multiple clones that use the same V and J. And you can see that the response is quite diverse. So we do have different V being used and different J's being used into different patients. And this will become important later. This is just the patients that didn't have any kind of metastasis. And those are the patients that have lymph node metastasis. And you can see that there is no difference. So for any patient that had a response, the response is quite diverse. So next, we wanted to see, okay, so the response is diverse, but is it shared between patients? So do we have the same TCR being present in multiple patients? So to do that, we look into the CDR3 sequence. So that is the amino acid sequence that falls exactly in the recombinant VDJ region. And by using that, you can look into the sequence, not using the whole sequence of the TCR, but just this sequence will determine the specificity of the TCR. And when we do that, and we look into the CDR3, What we see is that no, most of the patients don't have any shared CDR3s, both alpha and beta chain, with each other. So we only have, for example, here, patient 8 with patient 6, we have 2 alpha sequences and 1 beta sequence that one might be pairing with the other, and it could be that there is one shared between 2 patients. So this shows that the response is quite private. So the TCR that we find in one patient, we don't find in another patient. Just to confirm that, rather than just looking at exact matches, we also did clustering analysis, which means it's with glyph2, and we look into the sequences, and it allows for a little bit of amino acid changes between the 6 sequences. And even allowing for these small amino acid changes, we still don't have a lot of shared sequences, and most of the shared sequences are between 2 TCRs of the same patient. And this is not due to PCR error, because we do use the UMI, the unique modified identification, during the PCR before we start the PCR reaction. So we know that these sequences, they are actually, they are different. It's not due to just a mutation during the PCR reactions that we use for the TCR sequencing. But yeah, this shows that the response is quite private, and this could be due to differences in HLA typing. But most of these patients, they come from European origin. So half of the patients, they are HLA-2, and they do have very common HLA sharing between them. So another reason that this could be is that indeed we do have different antigens being presented by different patients, and that's why we don't have this shared response. Of course, we are looking at the circulating tumor-reactive T-cell response. So we know that inside the tumor, we will have what we call bystander T-cells, which are T-cells that are not specific for the tumor, and tumor-specific T-cells. So do we see any difference in the number of CD8 T-cells between the patients that don't have a response and the ones that do? So for breast cancer is a very cold tumor. So it doesn't really have a lot of CD8 infiltration, except for very few cases. So what we observed was like all the 3 patients that we had that had this very high infiltration, which is rare, were among the patients that had the tumor-reactive CD8 response. But more than that, we also looked into the difference between the tumor and the par-tumor in terms of infiltration of the T cells, because as I mentioned, one of the ways that the cancer can block the T cell response is basically blocking the infiltration of the T cells from the tumor. So, of course, the par-tumor tissue, normal breast tissue, is not very infiltrated with CD8s. So you will have a number higher than 1. But for the patients that have the blood tumor-reactive response, we do have a significantly higher amount of these CD8s inside the tumor. So as I mentioned on the very beginning, we did have 30% of the patients that didn't respond. And our question next was, what clinical characteristics could explain and separate the patients that do respond and the ones that don't have a tumor-specific response in the blood? So we looked into many, many different parameters, and the only one that came up as significant was the lymph node metastasis. So we looked into size, stage, many other characteristics, but the only thing that separates the patients is the lymph node metastasis. And what we found was the patients that did have the lymph node metastasis, half of them didn't have a detectable response. And that was true even when we look into the different histological subtypes. So the breast cancer not only have the molecular subtype, which we are working with hormone receptor positive, but it also has histological subtypes. So if you separate into the different histological subtypes, you don't see any difference. But we also see the same tendency that no matter what is the histological subtype, the ones with lymph node metastasis are the ones with lower response. And our first question was actually, are these patients the ones that have less antigens, and that's why they don't have a response? And that's actually not the case. So if we look, so that's the TCGA database, and if we just look of number of expressed cancer-testis antigens and the mutation count between the patients that do and do not have lymph node metastasis, this is not different. So we don't know exactly why this difference is happening, but it's definitely not due to a lack of antigens. So far, what we have shown is that the early stage tumor, so the localized tumor, it does have a very high circulating tumor reactive T cell response in the blood. And these have a very diverse repertoire. But once you have patients with late stage, you have most of these patients not having a circulating tumor-reactive T-cell response. So of course, we wanted to next look into what was happening inside the tumor. So for that, as I mentioned, inside the tumor microenvironment, usually the cells are very exhausted and they don't have functionality anymore. So to avoid doing any functional assay on the cells in the tumor, we actually did an indirect assay. So we looked into the tumor-specific T-cell sequences, the TCR sequences on the blood, and then we looked into how many of them fully match the sequences inside the tumor. And that would give us the tumor-specific T cells, no matter what was the functionality of these cells. So when we do that, both for the alpha and the beta chain, we do see a difference. So this is only the patients, of course, that have a tumor-specific response in the blood, which is only 5. But when we look into those 5, we still see a sharping decrease in the percentage of tumor-reactive cells inside the tumor. Of course, we want to see if that is specific for the tumor-reactive cells and not anything else. So we have done pathogen-specific specificity, so there is no difference in there. And also there is no difference in the total CD8 TOs. So there is no difference in the number of CD8s, and there is also no difference between the number of unique sequences, so how diverse the repertoire is. So it's really a decrease in the tumor-reactive response. So to confirm that, we actually used 2 other single-cell RNA-seq databases. So these databases, they contain analysis of all the cells inside the tumor of the patients, so single-cell analysis. So we selected only the hormone receptor positive, which are the patients that we are interested, and they had the lymph node metastasis information on them. So we can separate the patients that do and do not have for each of them, and we can filter to include only the CD8 cells. So we end up with that many CD8s for each of the databases. And how do we know if these cells are specific or not? That is another way of doing it in an indirect way, which is using this neoantigen-specific gene signature. So this paper has been published recently, and it shows what genes usually the cells that are expressed on these kind of patients, they express if they are tumor-specific T cells. So we use this signature to look for the CD8 cells that have this high signature, which means that they have a higher probability of being tumor-specific. And then we do the score of our individual cells. And we do that, we see exactly the same thing as we saw on our method, that the patients that without the lymph node metastasis, they do have a lot more cells that has higher neoantigen specificity score compared to the patients that do have the lymph node metastasis. We also did the frequency of the cluster with the highest score. So basically, we do have these UMAP analysis where you can cluster the cells in different clusters. And we got the cluster that has the highest, the frequency, the highest score for the neoantigen. And then we calculated the frequency of that cluster in each patient. And again, we see the same thing. So it really shows that in early stage tumors, so once you have a localized hormone receptor positive tumor, you do have a high circulating tumor reactive T cell response, and you also have a high tumor reactive tumor infiltrating lymphocytes response. But once you have patients with late stage with lymph node metastasis, you have lower circulating response, but also lower tumor reactive tumor infiltrating T cells. And that actually can explain why these patients don't respond to checkpoint, because patients on the early stage, when they have just localized tumors, they just receive the surgery and the tumor is gone. So we don't tend to treat these patients with anything else rather than the surgery. But at the late stage, that is when the patients start to need other type of treatment besides the surgery. And that is the kind of patients that are being treated on the clinical trials for checkpoint blockade. But if you think that these patients don't have tumor-specific T cells inside the tumor, there is nothing to release, so they will not be able to kill the tumor cells if they don't have tumor-reactive T cells to start with. And that actually gives us a hint of what kind of treatment that we can use for these patients. So these patients, since they do have antigens, as I showed before, it's not due to a lack of antigen, these might benefit from immunotherapeutic approaches that aims to replenish this response that they are lacking, either by vaccination or by doing a TCR-T or a CAR-T approach, where you inject the T cells that are specific directly rather than just a vaccine. The problem with these is that most of the vaccines, and especially the TCR and the CAR-T, they require the identification of either the antigen or the TCRs that are clinically relevant. And as I mentioned in the beginning, for breast cancer, it's very difficult to know what is going to be the antigen that these cells are specific. So now what we are doing is that we are trying to find those relevant tumor antigens and tumor reactive T cells to see if we can use that in the clinic. So we are using different approaches for that. One of them is trying to identify tumor reactive T cell clones that react to shared antigens. So antigens that will be expressed by more than one tumor cell. And the way that we are doing it is testing our clones against different cell lines and selecting clones that react and kill multiple different cell lines, because these would probably be specific for a shared antigen. So if you consider the cancer testis antigen, from these three cell lines, we can see that they share some of these antigens that we already know, but the antigens, which are the mutations, they're very specific for each patient. So we do believe that we might be able to find some T cells that will have shared specificity among different tumors that could be used therapeutically. We are also doing the other way around. So we are trying to look into, for example, the TCGA dataset for antigens that are more expressed in breast cancers, and then testing those antigens, those peptide pools directly into the T cells. So far, we have found some antigens, but none that were very broad. So rather than having like a more, so we prefer to do a more antigen agnostic way. So we don't know the antigen. We just find cells that are T cells that are specific for multiple tumor cells, because it's very, very difficult to find the antigen. And that was why this kind of study wasn't done before. And lastly, we do have, once we find those kind of T cells that are of interest, of course, we would want to know what exactly are the antigens that they're specific for. That is very, very difficult. But we have partnered with Ricardo Fernandez here in Oxford, and he does have this quite nice yeast library display that gives us a hope to try to de-orphanize this TCR. And how it's done? So you have this yeast library. So these yeast, they express MHC, and they will express a random peptide. Each yeast will express a different peptide, and then you do a binding assay with your tumor-reactive TCR. So you express your tumor-reactive TCR, and you bind it to your column, and you select only the yeast that binds to the column. Then you grow that yeast, and you do that multiple times until you have a library that is enriched for some peptides. So you can do the sequencing, target prediction, and peptide validation. So this is just one example of how it works. So you have the MHC expression on the yeast in here, and the TCR tetramer. So this is a tetramer of the TCR, not the MHC. Our TCR of interest. So you can see uninducted cells, of course, they will not bind the TCR, and they will not have any MHC on the surface. You do then start, so you do induce the library into your yeast. So they will start expressing the MHC. On round one, you usually don't see anything, but as you progress, you will start to see enrichment of cells. There are MHC positive, but also positive for the TCR tetramer. So they are binding the tetramer. And then you have to go to all this sequencing, targeting prediction, and peptide validation to try to find what was the peptide that these TCRs are specific. So with this, I just want to finish by answering the question. So what can they teach us when you do this analysis on the tumor-specific T cells? So we do, for example, have the tumor immunosurveillance. So when we look into the healthy donors, we start to find these cells that other people have found before. But the most interesting thing is there is a potential for developing prophylactic vaccines if we further study these tumor-specific T cells in healthy individuals. we could potentially try to develop a prophylactic vaccine out of it. So we also show how vaccination can induce tumor-specific T cells and why it might fail. And that is important because it gives you insight on how to develop better vaccine strategies, but also to predict which patients might benefit from the treatment. So I didn't show here, but we tend to see that the patients that didn't have a response before they start to receive the vaccine are the ones that live longer compared to the patients that already had a response when they start. We also can look into the presence of tumor-specific T cells in less immunogenic cancers. So these cancers are usually not studied in terms of immunology because they are very difficult to find an antigen to look. So when you use this more broad approach, you can start to see the presence of these tumor-specific T cells in these cancers, and you can study this to try to guide treatment selection and personalized treatment for these patients that are more difficult to treat with immunotherapy. And there is also a potential to find therapeutically relevant tumor antigens once you do this more broad approach that is antigen agnostic. You can use that to find cells that are specific, and then you try to find the tumor antigens rather than the other way around, which is quite difficult. And lastly, we do see that the tumor progression shape the tumor-specific repertoire, at least in hormone receptor-positive breast cancers. And that is very important because we can now start to understand which immunotherapeutic approaches might work for these patients. And we also understand how early versus late immunotherapeutic approaches might be different in different patients. And that's the main thing with by analyzing this tumor-specific T cell landscape, we can learn a lot into what kind of immunotherapy we could use for which patient and what would be the best immunotherapy to use, because each tumor and each patient is going to be different. So with that, I just want to thank. There is a lot of people to thank. So in red is the postdoc part, and in blue the PhD part. So now I'm in Tao Dong's lab. So I had huge help from Balraj and Ting with the experiments. So we do have Ricardo Fernandez helping with the East Library. So there is a bunch of clinical staff that helps with the patient, with collecting the patients. So we are very glad to have collaboration with them. We do have collaboration with the Oxford Biobank that helps us to collect these samples. And of course, the facilities. So we have fax facility and sequencing facility here. And of course, we want to thank all the patients, their family. So on the first part, I do mention that I was part of Barbota's lab, and I'm very glad for that. Patricia also helped a lot. We did have a big team on the glioblastoma that helped processing all these samples that were coming for the clinical trials. And of course, Federica Saluso, because without her, I wouldn't be able to start everything with her method. And of course, for the breast cancer samples, we had help from the Parallel Hospital. We have irradiation with the EPAN, and we also had help with Mary Sodaya from the cell. And with that, I want to thank and ask if you have any questions.
All right, thank you, Mariana. So we always give preference for the students first. So is there any question here? São Paulo, in Rio, or Uberlândia? If not, I would like to start with a question. Actually, there's a question from Gustavo Rocha. So Gustavo, if you want to open your mic. Gustavo? Well, do you have questions from students there, Bruno?
No.
So I'll go ahead and start with the question, Mariana.
Yeah.
Actually, you're mentioning both the early and late stage tumors.
Yes.
They have availability of antigens, right? But you don't have tumor infiltrating lymphocytes. So the difference is one of the differences on the tumor infiltrating lymphocytes, right? The circulating lymphocytes and tumor infiltrating lymphocytes.
So they do have the same number of tails, CD8 tails, but the number of tumor-specific ones is different.
Oh, I got it. I got it. I was wondering if there is some difference in the characteristics of those those lymphocytes on chemokine receptor or stuff like that. Are they able to get to the tumor? You know, what happens with them?
Yeah, so now it's the question, because we want to know why they don't have this response, right? So there is like actually two hypotheses that we have. One would be that the patients that didn't have, for any reason, like either they don't have the priming or they don't have the infiltration ability. The patients that don't have the tumor-specific T cells inside the tumor, they would be the ones that have progressive disease. And then when you catch it, they already have the metastasis. So that's one of the hypotheses. But another one is that they had the response to start with, but this response is then suppressed when the tumor cells reach the lymph node. And there is support for that, because they did find, there are a couple of studies, recent studies, that found that the lymph nodes that have Tregs on them, tumor cells on them, they have more Treg infiltration, and this Treg blocks the response. So it could be both things. We don't know yet, and it's very difficult to do temporal studies on these patients.
Right, and that's another question that I was thinking. I was thinking about it during your talk, and then in the end, you mentioned it like a potential for this mapping of the epitopes, right? Of the TCRs is about trying to develop protective vaccines. So my question is, do you know? I don't have too much knowledge on the literature on tumor immunology, but do you know about studies that have been mapping the responses to common or already described breast cancer antigens in people, in older people that did not develop tumor, you know, among the memory cells? Do you know of studies that are doing that?
You mean on the healthy donors?
Yeah, the healthy donors that they may have some kind of mutation during life, developed an effective T cell response, did not develop tumor, and then you have a specific memory T cell against those antigens. That would be, you know, a nice strategy to develop a vaccine. Do you know about studies like that?
No. So it's very difficult. They don't even have studies with cancer patients. So that's the main thing, that it's new in here, that we can actually go deep into what is the antigen and what they are responding to. Because once you try to find, oh, there is this antigen that I know that is expressed, and then you try to find the T cells, you don't find it. And then you just say, oh, this patient doesn't have any T cell, any specific T cell, but it does. It's just that you are looking at the wrong antigen, and we don't know what are the antigens that actually give responses to breast and to glioblastoma. Actually, those are the two tumors that we don't really have antigens known. So we do have some, but they are very restricted. Most of them, they're less than 10% of the patients express it, and less than 5% of the patients actually have T cell specific for.
I got it. So it's hard to find in patients, imagining healthy people.
Yeah.
All right. So there's a question from Bruno, then one from Barbuto, and then there's a question in the chat too. So go ahead, Bruno.
All right. Thank you, Diego. Thank you, Mariana, for a very, very nice talk. Very, very interesting. I don't know if I missed something about your patients, if they are treatment naive, if they have received any sort of treatment beforehand, before you collect these lymphocytes, and wondering if chemotherapy would actually change the profile of the tumor-reactive T cells. And also, regarding the tumor-infiltrating lymphocytes, the immunocheckpoint inhibitors, they have some chance of increasing the number of tumor-reactive T cells that you get there, because since the lymphocytes are already there, they are probably, even if in low numbers, they are probably being inhibited, right? So if you block these inhibition, maybe we could get more T cells from these tumors and eventually use these T cells as basis for CAR T or something like that.
Yeah, so the problem is like they don't have tumor-specific T cells inside the tumor. So in melanoma, you do have a lot of tumor-specific T cells that are infiltrating, and then when you use the anti-PD-1, you release those cells and they can kill. So I am actually studying the triple negative now that are being treated with checkpoint to see if they can actually also generate a new response in the blood, you know, that can infiltrate. But that's another thing. So far, what we think happens when you use the checkpoint is the release of the cells that are already inside the tumor.
Right.
So you need tumor-specific T cells. So the first
yeah, but you do have a little bit, right? So I was wondering if these little bits may get a little bit bigger if you do the immune checkpoint inhibitors.
It's possible, but you can see that the response for these kind of patients, it's really, really low. And when I say really low, it's like less than 1% of the patients respond. So probably those are the ones that have the highest, you know, the highest response.
Okay, got it. And the first part was about chemotherapy and how chemotherapy was.
So that's a great question. So I forgot to mention, but all my patients on the study here in Oxford, they are treatment naive. And they go to surgery first. So I get the blood and the tumor when they are treatment naive. So the only kind of treatment that they can have is letrozole, which is one of the blocking of the estrogen receptor. But that usually doesn't affect a lot of the response. And I only had 3 out of 23 patients that had that treatment before. So most of them are treatment naive. And we didn't see any difference on these 3 patients that had the letrozole. But yes, chemotherapy can affect the response. Especially because it does kill a lot of cells that are proliferating, and it can also kill T cells. So we tend to avoid that. But for example, for the other study that I mentioned that we are doing now with the triple negative, they have both chemotherapy plus checkpoint inhibition. And one of the questions of the project is actually to see if the chemotherapy makes it better or worse, because at the same time that the chemotherapy will release antigens that will be able to activate more T cells, it can also kill the cells that are being activated, right? So we don't know what is best.
Right. Thank you very much.
Right. So we have a question from Barbuto. You want to go ahead, Barbuto? We can't hear you.
I always suffer with these controls. Very nice talk, Mariana. I liked very much. Thank you for reminding me of it. My question is just a suggestion. You studied early tumors that have already metastasized to lymph nodes, but what happens when the tumors really get late-stage tumors, really, the ones that are metastasized at distance? Did you check? Do you pretend to check? Because then the response may change completely.
It may. The problem with that is those patients, they go to a lot of treatment before the surgery, and they don't even get the surgery. So it's a bit more difficult because, so these patients, they will have chemotherapy and other kind of treatment.
It will be very, or you can, the patients that recur, actually the patients before they start, because they are doing something, and then they state the disease recurs. You have to consider even the presence of chemotherapy, because that's that's the real life. The patients will have that. Do they have the cells? It would change a lot how we understand it.
Yeah. Oh, definitely. And that's, again, another thing that I'm doing on the other project, because we do have neoadjuvant and adjuvant checkpoint blockade for breast cancer. So we do have patients that are receiving it before the surgery, which are the ones that have smaller tumors and the localized tumors, and ones that are receiving it and already metastatic, and they behave completely different. So it could be, but again, it's like one already have both of them have the tumor in there when you start a treatment, but they are completely different in terms of what kind of treatment they get. Usually the late one receives PD-L1 while the early one receives PD-1. So it's different.
Okay, thank you.
All right, so I would just go with 3 questions that are in the chat, and then I can open to Renata, and I see Miriam with her hand above too. So First question in the chat is from Maria Fernanda Santos Lopes from Federal University of Uberlândia. She said, thank you very much for the nice talk. What do you see as the main obstacles in translating these findings into clinical applications?
There is a lot of obstacles, I would say. Finding the T cell and the antigen now is the most difficult one. So now that's what we are looking for, to see if we can translate that. And yeah, I would say that as immunologists, we usually, so the clinicians, they usually tend to get a treatment that works in one cancer and that they try in different cancers, right? And we now know that the same treatment is not going to cure all kind of cancer. So I think understanding exactly what is going on with the tumor-specific T cells in each patient is the most important step, because not only between different tumors, but even within patients that have the same kind of tumor, it's different, the evasion mechanism that they have. So if you can understand, and if you can do it in a way clinically that we can evaluate those cells in each patient, we would probably be best, we would be in a better place in terms of trying to treat each patient individually.
All right, so another question in the chat. Let me just roll it down here. I can see it now. So, hi, Mari. This is from Barbara Gustinelli. Great talk. As you know, Barbuto always stresses about the importance of CD4 T cells in tumor response. I might be wrong, but some of the results you presented were more focused on CD8 specific response than CD4. Have you seen significant difference also in CD4 populations?
That is a very, very, very nice question. So my supervisor in here, she works more with CD8s. So I did have to focus on the CD8s in here, although I do have some CD4 results now that we are putting together. But yeah, I think the CD4s are very important. I believe in the same thing as Barbuto believes. But it's difficult to convince the broad community that the CD4s are important. So we do step by step, but we do see some differences in CD4s, but it's not the same thing. And they are way more difficult to study, actually.
Another question from the chat before I open to Renata and Miriam. Great talk, Mariana, from Luis Felipe Cruz. I have one question. How is current research dealing with exhausted CTLs that are immunomodulatory? I was wondering if you could talk a bit more about it.
CD8s or CD4s? Because the CD8s, basically what they do is the checkpoint inhibition. And what the field is trying now is to find other kind of inhibitory molecules that are being expressed. So there is a lot of work on the phenotype of these exhausted cells, because we do have the concept now of terminally differentiated exhausted cells and progenitor exhausted cells. And that's more in mouse, but there are some work coming now in humans that you do have, that's the checkpoint, they actually reinvigorate those progenitor exhausted cells, but not the terminally differentiated cells. And there is a lot of interest in the field trying to find markers for these two populations and how they differ with each other.
Renata, do we want to make your question?
Yes, thank you. Hi, Mariana, very beautiful work. Congratulations. So, yeah, my questions are also about exhaustion. So, in the first one, the glioblastoma project, did you observe higher proportion of terminally exhausted cells? Did you check these? And do you associate the increase in Th17 as a cause or a consequence of tumor growth? I mean, could you have more Th17 as a consequence of inflammation, and then it's not associated as a cause? And in my second question for the second project is also associated to exhaustion, because you propose the use of CAR T cells, as you don't find the antigen-specific cells, but your expectation is that these cells could also get exhausted in this tumor microenvironment.
Yeah. So usually, and I haven't done a very broad analysis to say for sure, but at least for breast, it's more known that these cells are actually not many cells inside the tumor are exhausted. And I have a feeling the glioblastoma is going to be the same. It's because it also doesn't work a lot with the checkpoint inhibition, you know. So these tumors, they tend to not to work as different evasion mechanisms rather than the exhaustion part. So the cells don't even get to the tumor. I don't know exactly for the glioblastoma, but for the breast, that seems to be true. For the Th17, I do have a lot of work that I have done that we still have to publish during my PhD on the breast cancer. So the tumor-specific CD4 T cells on breast cancer, there are a lot of them that are Th17. And we have a couple that with neutrophil infiltration on these tumors. So the presence of these cells with neutrophil infiltration. And yeah, so we are working on this story, but they are related. And the patients that have more of it, they have more neutrophil. So we think it's bad. So we saw that on the glioblastoma patient, and we saw that on a cohort of breast cancer patients. And the last question, sorry.
No, that was the question, the Th17 index. Oh, and if you, okay, you answer about the CAR-T and the breast because you don't expect them to get exhausted.
I do expect, so once you start adding the cells, then you will probably have exhaustion being a part of it. So I do think if you start to vaccinate and you start to have this big response, the tumor might change, the way that it blocks, and it might start to have the exhaustion, right? Mm-hmm. If that happens, there is like now a huge, the field is going through like these patients that don't have response to try to do vaccine plus the checkpoint.
Combine it.
So you already combat, any kind of exhaustion that can happen on these patients.
Perfect. Thank you, Mariana.
Right. So there's a question from, I see Miriam. Bruno raised hand, but Miriam is there.
Yeah, so thank you. I'm using Bruno's name, so it's a bad question. It's his fault. So I'm joking. So Mariana, thank you for your talk. Very, very interesting topic. I wonder if you were thinking or if you thought it could work to do like a proposed study where you get a cohort of BRCA mutated patients that did not develop the tumors yet, and you follow their T cell specificity to see if you find settings where they appear more or less because of the data you showed on the metastases, or basically the positive lymph nodes, that I thought was related to having, when you have more tumor-specific T cells, you have less positivity on the lymph nodes, right? So I wonder if you're thinking, or if you thought it could work something like that.
Yeah, so there is a lot of projects now that I want to do that requires like following the patients for longer, like this one. So that's one of the ones that I was thinking, because the BRCA one, they do develop earlier than other patients. So you could potentially follow these kind of cohorts. The problem is like when you're doing a PhD or a postdoc, it's just like a short period of time, and it already takes very long time just to collect the samples to start the study. So I haven't tackled those kind of questions yet, but once I have my lab, that's the kind of questions I'm definitely going to try to follow patients for way longer now, to see if these patients develop or don't develop. And the BRCA is a very nice cohort to have.
Okay, thank you.
All right. So there was another question in the chat, but the question is gone. So I think we're done with questions today. I would like to thank you a lot, Mariana. It was a great talk. You saw a lot of people ask questions, so that's a sign it's a good talk. Thank you very much for accepting our invitation, and I see you guys next week. Thank you.
Yeah, thank you for having me.
Bye-bye.